Hi everyone, this is Ramalinga Prasad Kuppa. Welcome to my channel Pharma World. Today's topic is Analytical Procedure Life Cycle Approach as per USP Chapter 1220. This chapter considers the development, verification, and validation activities across the entire life cycle of an analytical procedure. The life cycle approach emphasizes the importance of sound scientific principles with risk assessment at every stage of development and qualification. Let us see more details. Life cycle approach. It is across the entire life cycle of an analytical procedure from development, validation, changes in the procedure during revisions, evaluating the adaptability for changes in compendial revisions till the product discontinuation. So, the approach is to focus on the analytical method from start to finish. These are potential aspects that can take place in a life cycle approach. The validation parameters may be revised based on the impact of the changes in the method. The changes could be triggered by changes in compendial specifications and methodology changes. As said earlier, this attitude and approach should be adopted till complete discontinuation of the product and its related services. The procedural life cycle approach emphasizes the importance of sound scientific approaches and quality risk management for the development, control, establishment, and use of analytical procedure. Scientific evaluation is necessary to establish a suitable test method. The risk assessment with statistical evaluation will provide very useful inputs for the development of the method. The controls may include system suitability parameters, sample preparation, standard preparation details, and any other critical test parameter controls, etc. Total error and measurement uncertainty should be focused for the proposed analytical method. Accuracy and precision are part of the total error. Accuracy is defined as closeness to the true value, and precision is the closeness of the replicate values. It is the measure on how much is the random variation in the results obtained. Measurement of uncertainty can also be used. Method development. Quality by design, that is QBD, concepts as prescribed in ICH guidelines should be used for development of the analytical method. This is a very important concept to understand. Quality is built in the design itself. The right design with appropriate controls will yield the successful method. ICH guidelines describe the requirements in an elaborate way. Developing the design space is one of the key requirements. The procedure life cycle includes analytical target profile ATP and three stages. ATP defines criteria for the performance characteristics. It is linked to the intended analytical application and the quality attributes to be measured. The reportable value generated using a qualified analytical procedure provides the basis for key decisions regarding compliance of a test article. Yes or no to the results for compliance. Overall focus of having a procedure with acceptable bias, that is accuracy, and precision should be the part of ATP. Stage 1 focuses on procedure design, stage 2 focuses on performance qualification, and stage 3 focuses on ongoing performance verification. Next step is to develop these stages 1, 2, and 3 for monitoring the performance verification. Procedures should be developed to have levels of bias and precision that are consistent with the ATP criteria. Levels of accuracy and precision distribution across the center can be achieved by statistical evaluation of data with relative standard deviation of replicate results. Using statistical tools, a maximum allowable combined bias and precision should be determined that provides appropriate performance for the procedure. So the statistical tools are very important to establish the ATP criteria. A decision rule should be used to define the maximum allowable combined bias and precision. Based on the evaluation, a decision rule may be developed. This becomes the basis for taking a decision on compliance of the article to the specifications. A simple decision rule is to accept within acceptance specification zone with lower acceptance limit and upper acceptance limit. Obviously, the results within the lower and upper range of specifications can be considered for taking the decision for compliance. If the specification acceptance criteria are well within a safe and efficacious range, the decision is easy to pass or fail the product. This stage is simple to understand. When there is no element of doubt on the data spread, the decision is clear and unambiguous. In cases where reported values are closer to the lower and upper acceptance limits and part of the reported distribution is out of acceptance criteria, that is indecision zone, a guard band should be used. This is important. In certain cases, some part of the distribution may be outside the upper limit or lower limit of the specifications. In this case, it is difficult to decide on compliance. This is an indecisive situation. In such cases of indecision zone, additional guard band is necessary. In such situations where additional risk reduction is desired. Decision rule using indecision zones should be based on the total analytical error for safe and effective range reportable. When the reportable value is within the indecision zone, a predetermined supplementary sampling and testing plan and decision rule may be used to determine conformance. Let us see some details of stage 1 of method development. Procedure design should be such that any technique that is capable of meeting the ATP criteria can be selected, which should include relevant information linked to the operational requirements such as instrumentation type, setup and sample preparation, reference standards, reagents, etc. The draft test method should focus on these items carefully. during the development stage. It can be developed on chemical structure of the article. Classical chemistry test principles may be selected if the functional group is identified unambiguously. Instrumental techniques may also be selected for many organic compounds. Most of the organic compounds have absorbance in UV range between 200 nanometers and 400 nanometers. For such compounds, HPLC methods are suitable. The solubility of the compound, its other characteristics may also be considered while designing the method. The main purposes of the procedure development are to identify conditions that minimize or avoid bias, to optimize variability, and to establish robust operating parameters that have the potential to meet the ATP. Any method should be able to comply to these basic requirements. Otherwise, the method is not suitable and cannot be validated. The method should be evaluated using the QRM principles, that is ICH Q9. Each parameter should be included in the risk assessment matrix. Each parameter should be evaluated for risk. FMEA or FMECA tools are very useful. Either of the two tools are okay. FMEA classifies qualitative risk and FMECA classifies the risk quantitatively with a score. QRM process is to evaluate the proposed procedure conditions and identify appropriate controls on the analytical procedure that will ensure the procedure meets ATP. The result of risk assessment will help in developing the necessary controls of the test method. The robustness is another parameter to establish for the analytical method to establish the capability of a procedure to remain unaffected by small but deliberate variations in parameters and to maintain suitability during normal range. Robustness is a parameter to ensure that the method works well even if there were accidental unintentional variations in the controls. Understanding of the overall sources of variation contributed by the procedure should be established. The variations are best evaluated by statistical variability test with the data sets. Analytical control strategy ACS is another important aspect to establish system suitability tests and other environmental or procedure controls needed for the procedure to meet ATP. There is a general thinking that the system suitability tests are relevant to HPLC or GC analysis only. In reality, the system suitability is a overall control strategy for critical parameters of any test method to ensure that the method works as designed and expected. Critical procedure attributes identified during development should be controlled and their acceptable conditions, materials, or criteria should be explicitly specified in the procedure. Critical procedure attributes include selection of appropriate tools, glassware, grades of the reagents necessary like AR grade or HPLC grade, spectroscopic grade, etc. All these items should be explicitly prescribed in the test method. If it is a wet analysis method, it may be necessary to prescribe explicitly the requirements of glassware, including the selection of burette or pipette with necessary least count scale denomination. The strategy of stage 1 with its associated initial control strategy should be the input to stage 2 of the life cycle. This initial stage information may be used for the next stage 2 of life cycle development. Let us see the details of stage 2 of the method development. Stage 2 involves qualifying the developed method for suitability on routine basis. Stage 2 involves qualifying the method developed in stage 1. Qualification may be considered as a trial run of the test method before going for full validation. Once the procedure is established, the procedure is ready to undergo analytical procedure performance qualification APPQ. APPQ is full validation of the test method as per USP or EP or ICH Q2 or Q14. The information will be more or less same in all the guidelines. The protocol and study design should at least include description of the procedure, the parameters like accuracy, precision, range, specificity, calibration model, quantitation limit, robustness, etc. The validation should be done as per USP 1225 and verification as per USP 1226, etc. USP chapter 1225 prescribes detailed methodology for validation with these parameters. Verification activity is done for already validated test method. The compiled methods are published only after validation studies and approval by the agency experts. That's the reason why only verification activity as per USP 1226 are carried out for compendial test methods. After successful qualification, the APPQ, the data generated should be documented. The data generated should be documented as objective evidence of validation. This is a basic requirement as per GMP and GLP. Lastly, let us see the stage 3 of method development. Stage 3 includes routine monitoring of the analytical method and evaluation of the performance of the procedure. In this stage 3, huge analytical data is collected to evaluate its performance. Routine monitoring includes evaluation of data generated by statistical tools. The data evaluation is done by statistical methods scientifically to establish the performance of the test method. The analytical control strategy can be used as a source for identifying the relevant attributes used for ongoing monitoring. The evaluation may identify some of the critical parameters that are monitored to revise the test method if necessary. Control charts including X-bar graphs, MR graphs, Pareto charts, scatter diagrams may be useful for trending the data generated. These tools provide the information on distribution of the data. Let us see how the changes to the established method can be handled. Changes to analytical procedures may be needed over the life cycle and can be prompted by several events, including needed changes identified through a routine monitoring program, the adoption of new technology, or changes to the ATP. The data generated as explained above. The data is useful for review for any necessity to revise the existing method for betterment. I hope that this information on development of analytical method is useful for developing any new method or modifying the existing method. Thanks for watching. For more videos, Please do subscribe, like and share. Also, please leave a message in the comments box for any further support. Thank you.